Good morning, everyone. My name is Jonathan Knittel, and I was working this past summer in Professor Venturis's lab, and my project was called the MIF characterization of internally tagged variants. So a little bit about myself. I grew up in Discovery Bay, California, which is about 20 minutes west of here. It's not too far. I'm a biochemistry major. I'm graduating in the spring of 2022. And I came to UOP because it was the only school that I found that would give undergraduate or give freshman research opportunities. And I also saw the Stauffer as a way of kind of getting more research out of what I've made in the last year and a half. And so a little bit of an introduction about enzymes before we move into the project. Enzymes, which are marked as E, interact with a substrate S, and we'll change it into a product P. And so you can see here along the bottom, the enzyme is taking in the substrate, and then there's an enzyme substrate complex here, and then the product is removed from the enzyme. And by studying these reactions, we can tell the mechanism and as well as how inhibitors will affect the rate of the reactions. So what is kinetics? Enzyme kinetics measures how fast a reaction occurs. And so this here, the E plus S going to ES, making E plus P, is just a wording representation of the previous picture. And that's the Michaelis-Menten model for kinetics, and that's what we focus on when we do kinetics. And so we want the initial rate of the reaction. So here you can see there's an initial rate, and this shows the formation of the product on the y-axis. If you take these initial rates for different concentrations, you were able to make a graph that looks like this at the bottom, where you have the max rate of the reaction over the different substrate concentrations, and it also has half Vmax and Km, which we'll talk about in a minute. And this on the right side is the machine that I used for kinetics this past summer, sorry. And so the research objectives for my project, so macrophage migration inhibitory factor, or MIF, as I'll call it from here on, is known to bind and activate the cell surface receptor CD74. And MIF is also a cytokine, which means that it kind of mediates reactions in between cells and different cell types. So what we did was we took 11 MIF variants were made with histidine tags. And so you can see the pictures on the right side. You can see where the histidine tags were placed. And they were placed in between the N and C terminals of the protein to keep the functionalities of it, but they're on surface exposed parts so that it can be used for purification. So when it's purified, we use affinity chromatography, and that's the pictures in the bottom. This is the column, and the column is hooked up to the active. What you do is you load your sample, and you'll run it through the column, and the column will pick up your protein, and then you can get rid of the junk and dilute your protein after. So the objective was to understand the impact these tags will have on the purification process and the functionality. And so here is the kinetics data from this past summer. And so there are a couple definitions on the left side. Vmax is the maximum velocity when every enzyme is interacting with the substrate. Km is just the substrate concentration for half of Vmax. Kcat is how much substrate is turned into product by the enzyme. And so in this case, it's per second. And then if you do Kcat over Km, it just tells you how efficient your enzyme is. And so from this list, we wanted one that was similar to wild type MIF that's shown at the top here. And so we chose Q24H, Q25H, Q28H, because the Km is 1.4, which is similar to wild type as well as the Vmax. And then we were also able to eliminate the bottom 3 mutants that are shown, because if you look at the Kcat and Kcat over Km, they have almost no activity compared to the rest of them. And this is a representative graph of the kinetics I did. So like the previous picture, this is just one that I made after the analysis. You can see the different HPP concentrations along the bottom. And the impact of this research, the position of the tag will tell us information about the properties of the protein. And the findings will enhance MIF activation of CD74 receptor, and it'll serve as an example for other proteins that have similar structures that we can also use tags for purification. So there's other family members in the MIF superfamily, which includes DBT. And then the personal impact that it had on me was after being at home for a year and a half, Stauffer helped me get back in the lab to work on projects, and it also taught me how to design experiments as well as analyze and interpret data. And so for future plans for the experiment, the best mutant, so the one that I showed before, would have further experiments for their functionality. And so circular dichromism is the first graph shown at the top, which just shows if it folded correctly and if it's the right shape or if something happened along the purification process. And then crystallization, as well as in vivo studies. And so crystallization, and then the mouse is shown for in vivo. And then for my personal future plans, in the future, I plan to get a PhD, or I get a PhD in neuroscience in LA. And I want to use these skills and techniques that I learned during the Stauffer to get there, which includes a little bit of protein purification kinetics. So I would like to thank the John Stauffer Charitable Trust, as well as all the other donors. And I'd also like to thank Zara Bukhari, who's my grad student. She's here in the back, as well as Professor Tetomerase and the chemistry department, as well as the whole of the University of the Pacific, as well as my lab mates. So I'd like to open the floor for questions.
Yeah, I have a question.
Was there anything in your research that surprised you?
When I was doing the initial, or let me go back to the picture. So when I was first doing the runs, like the first graph you get kind of looks like this, and I was concerned for those 3 mutants that we eliminated because the machine just read over for them very quickly. And so since that was the first time I'd seen that, I wasn't exactly sure what that meant. And then once I done the rest of the data, it was just that they're not very active.
Well, I want to check this. This audience, can you hear this well? Question? All right, okay, so they are here well. Yeah.
What are the consequences of your finding?
What do you mean consequences?
What are you determining in regards to how it's going to play out when you get down to another facility? You have found certain structures, how they're going to go. How are you going to use them when you move forward?
So moving forward, what we want to do is take, because there's other proteins that are similar in structure to this, like that have a similar shape. And so what we want to do is, because some of those don't purify as easily.
Right.
And so we want to add these tags to those to hopefully make them easier to purify. And also make sure that it won't impact the structure of it, because then you can't use it to purify the
Complicated.
protein.
Okay, so yes, one more question.
So when you were picking, you can go back, maybe to your table. When you were picking the,
This one?
yeah, when you were picking that one in the middle,
This one?
because, was it the, K-cat that you, I'm sorry, I'm old, so I'm having trouble seeing. You picked the one with the 1.4?
Yeah.
That was close to the wild type?
Yep.
Why did you pick one that was close to the wild type rather than like a better K to K catalytic efficiency?
We chose this because we wanted it to be similar to wild type, just to prove like a purification platform that can then be used elsewhere. Because we didn't want to really impact the enzymes themselves, we just wanted it to be able to get them out easier.
Okay.
All right, thank you, Jonathan. So once again.
Hi everyone, my name is Jasmine Vargas. Today I'll be sharing what I did for the Stauffer this summer. I did research in Dr. Pantoris's lab. We worked on a project called Functional Insights into the D-dopachrome Tetomerase C-terminal, and I primarily focused on protein purification. So first of all, just a little bit about me. I'm a fourth-year biochem major and a transfer student from Delta College. I chose to transfer to the University of the Pacific mainly due to its good reputation as a local school and for its location. I live in Manteca, so it's very close to home and an easy commute. So first of all, what is D-dopachrome Tetomerase, otherwise known as DDT? DDT is a pro-inflammatory and tumorigenic cytokine that binds to the cell surface receptor CD74, and this binding is associated with many inflammatory disease and cancer. DDT is one of 2 human members of the MIF superfamily, and while DDT is similar to MIF in that they both bind and activate CD74, DBT is not well studied and is poorly understood. So for our research objectives, we wanted to determine the role of the DBTC terminal, which is pictured as bright blue tails here in figure 1. We would do this by expressing, purifying, and characterizing wild-type DDT and alanine mutants. We chose to mutate 5 key C-terminal residues listed here in figure 2. So we would mutate each of these individually to alanines, pictured here, to determine their function. As I mentioned previously, I worked on protein purification. I did this using the Active Pure, pictured on the left here. This is a system used for fast protein liquid chromatography. The 2 types of protein purification I worked on, one being Q-Separase anion exchange. This is used to separate proteins based on their charge, and the second being size exclusion chromatography, which, as the name implies, it separates proteins based on their size. And here are just some of my results. This is for the F116A DDT mutant. This was after running it through Q-Separase. Here on the chromatogram, I'm interested in the peaks here shown in blue. So I ran these individual fractions on a gel. The gel pictured here below is a way to visualize proteins based on their molecular weight. We know that DDT is a 12.5 kilodalton protein, so based on this ladder, we knew that our protein of interest was here in fractions 23 to 26. And we decided to further purify this protein using size exclusion to remove some of the contamination that we see here in lanes 25 and 26. So here are the results of that size exclusion. Once again, on the chromatogram, I'm interested in the peaks in blue. I ran them on a gel, and we were able to see that our protein of interest was here in fractions 22 to 27. We were able to remove most of that contamination. So from here, the protein is purified, we concentrate it, and we store it for future use. So as for the future plans for those proteins, we plan on running kinetic experiments. We plan on doing circular dichroism and doing protein crystallization to further learn about the DBTC terminal. And as for myself, I'll be graduating in the fall with my bachelor's in biochemistry. And I plan to attend Pacific for grad school in the spring, where I'll be pursuing my PhD in pharmaceutical and chemical sciences. For the research impact, by learning more about DDT in the future, we can help develop potential therapeutics. We would do so by creating selective DDT inhibitors that would block the activation of CD74. With this, we could help treat inflammatory disease and cancer, such as those listed here on the right. DDT inhibitors could work on multiple sclerosis, pancreatic cancer, kidney disease, and cardiovascular disease, just to name a few. And for the personal impact, the Stauffer gave me a lot of hands-on lab experience. I was able to learn new research skills, and I feel like participating in the Stauffer prepared me for what to expect out of grad school. And finally, being a part of the Stauffer, I was able to enjoy the campus in person, after being online for a year and a half due to COVID. And for my acknowledgements, I'd like to thank the John Stauffer Charitable Trust and donors for this amazing opportunity to do summer research. I would like to thank my PI and mentor, Dr. Pantoris. I'd like to thank the grad student I was working with, Andrew Perkins. I'd like to thank my lab mates, and a thank you to the University of the Pacific and the chemistry department for their support. That concludes my presentation. Let me know if you guys have any questions.
Wonderful presentation.
Thank you.
Now, questions? So anyone on Zoom, you can unmute yourself, just ask questions. I think we can hear. I think we should be able to hear. Or you chat, put down chat, I can read. Yes, Professor McCall.
So you may have said this, I just might have missed it. If you go back to any one of your gels,
Yes.
that one's fine. So what's the significance of that horizontal line? Is that like some threshold that you're using to cut off that you're seeing
Are you talking about this horizontal?
on the regular plot, on the physical, so like regular.
Oh, on this, oh, you're
So
talking
you
about
see the horizontal line on the top?
Oh, yes, this is the conductivity, so it's a measure, I believe, of the salt that we use for this purification. We did elute this protein at 15% of 20 millimolar tris HCl, 1 molar sodium chloride.
So I have a question.
Yes.
I do a lot of chromatography, but I usually do small molecules. On your previous slide, the one with what was the difference between this chromatogram and the next chromatogram?
So this chromatogram, as you see here, this peak is not very well defined, which, as the gel proved, there was contamination here. I mean, we can't tell too much just based on the chromatogram. We can see more based on the gel.
But I see that your conductivity changes here.
Yeah, our
Other than the other
conductivity changed here. Actually, I believe for this specific protein, we did do a gradient. So we went from I believe it was 10%, 1 molar salt to a 15% 1 molar salt. At that 15% is where the protein eluted out. And also, as a note, this is kind of a zoomed in version of the chromatogram versus this chromatogram. So you're just seeing more zoomed in, the more well-defined peak here correlates with the more pure protein. And here you're just seeing the zoomed in portion of kind of that
But is it
15%
the same graph?
It is not the
It's
same.
a different
graph.
experiment.
Yeah, so this is it's the same protein, the same mutant, but this is a Q-sucrose. So again, purifying based on charge.
Okay.
And this is the size exclusion, purifying based on size.
Very cool. Thank you.
More questions? Okay, so we can leave library class for during lunchtime. All right.
All right, thank you, everyone.
Hi, good morning, everyone. My name is Kara Talbot. I worked under Dr. Carlson, who is up here kindly helping us today. So my project is on how small molecule gene modification alters fungal natural product expression. Sounds kind of complicated, but I'll make sure to break it down for you guys. Next slide. So a little bit about me. I grew up in West Sacramento, so just a little hop away. I dreamed of attending the Pacific here ever since I did a little tour here in 7th grade, so it's a dream come true. I'll be graduating with my BS in biochemistry this Friday, actually, and I'll be starting my master's program in the pharmaceutical sciences this coming Monday. Very exciting. So a little bit of background. So our lab is very interested in aquatic fungi and the natural products they produce. A natural product is just a molecule that we find that it makes that we're very interested in, that usually has bioactivity that can like fight these antibiotics or maybe another antifungal. So super interested in that. But the production of natural products in fungi is tightly regulated, just like it is in humans. Like if it is silenced, it won't be made. So what we tried to do is induce the expression of new NPs or new natural products. So the fungal cultures were treated with DNA transcription modifiers, which I'll explain more on the next slide. So just a little bit of background, seems confusing. But the first one is just open DNA, which allows the DNA strand to be transcribed so that new products and proteins and little molecules can be made. The next step is DNMT, which is DNA methyl transferase, which is just those little pentagons. And when they come in, they add on the red dots. And the red dots are methyl groups, which means once a methyl group is added, so DNA is methylated, it can no longer be transcribed. And then a further form of DNA modification is the HDACs or histone deacetylase inhibitors, which are shown in yellow, which then tighten back up so that the DNA is extra closed and nothing can really touch it. So what we did is we did 2 different treatments. So we used transcription manipulators of 5-azacitidine, which is the DNA methyl transferase inhibitor or DMNTI. We also used sodium butyrate, which is a histone deacetylase inhibitor or HDACI. And this targets the DMNTs, stopping that, so we no longer have the red groups on there. So we can use the the now open DNA to hopefully make more natural products. Same thing with the sodium butyrate. The sodium butyrate attacks HDAC so that it no longer can be on there, and we continue to try to make new products. So one of my findings, LBC1, which is just one of the species of fungi that we were looking at. So this graph, I know looks confusing. This is HPLC, or high performance liquid chromatography, which is just us running our sample over a column, and it just dilutes out, and it just shows us spikes at different concentrations of UV absorbance. So what I really want to focus on is those two peaks. So if we look at those two peaks, those are new peaks. That's very exciting. It means our treatment must be working. We might have found something new. So if we zoom in, and you can very clearly see that it does not appear in the control or the two lower concentrations of our treatments. And so for our other one is SS781B, which is just another species that we ran these treatments on. Once again, same HPLC graph, but this one, all of our treatments showed a spike, except for the control, which is in the center, missing the spike. Here it is zoomed in, as you can clearly see, spike right there, but missing the spike in the control. So that means something possibly new could have come out of this. So our impact is it expresses previously silenced biosynthetic machinery in order to isolate new natural products not seen under normal conditions, right? So we're hoping that now that we opened up the DNA, we can make something new. If you don't know, that is the molecule of penicillin. Penicillin is a natural product produced by fungi. So that is really what our lab is trying to do. So our next steps would be to screen the extracts for antibacterial and antifungal activity induced by the treatment. So doing that, and then characterizing novel molecules, which means from those peaks, we'll try to grow up the experiment, optimize it so we can get the most of that product that we can, and then run LCMS, which is liquid chromatography mass spectrometry, which helps us identify if it is a new molecule that's found or a novel molecule, which would be super exciting because we can do more research into that. And so as I go into grad school, I'll just continue to optimize this technique and apply it to other fungal isolates. And so I would like to say thank you, everyone, for allowing me to have this opportunity. Especially thank you to the John Stauffer Charitable Trust and donors, the UP Chemistry Department, and from left to right, Michelle in the back, one of our grad students, my lovely PI, Dr. Carlson, Savannah, and Carolyn, who have greatly helped me get back in the lab after a year and a half of not touching any instruments. So I really appreciate everyone who could be here and everyone on Zoom. Thank you very much. And any questions?
So what's the Oh, great job describing epigenetics. I loved it. Such a cool trick. Do you have any controls to make sure that the new molecule is not the actual drug or some metabolite of the drug you're treating the yeast with? Or not yeast, sorry, fungus. I guess they're fungus, but the fungus.
So for this one, we were just trying to screen, but since it doesn't show up in the control, that kind of peak.
What's the control?
So the control was just, so each set just had control where it was untreated by the molecules. So is that. This one's just,
Can you show us your chromatograms when you're explaining this answer?
I absolutely can.
And can you also point out to everyone which one was the control, which one were your treatments?
So the top two and bottom two were our treatments, and the middle one is the control.
So Dr. Harrison is asking, how do we know that that's not the molecule we use to treat our cultures with?
For that one, I would like to look more at this one. So if you're just asking about like new peaks, both the new peaks should be the same in both, right? So since we had the treatment the same for the top two and the bottom two, just different concentrations, where for that not showing up in the top one, it shows us that the thing you're treating with really didn't enter into it.
Do both of your treatments have the same structure?
No.
Right. So if you're treating the same retention time, and Dr. Harrison's asking, is that the molecule that we used to treat with? Is this the molecule we used to treat?
No, it is not.
Awesome. Great controls. Controls are so important. So.
I'm curious, do you have any expectations as to what type of new molecules might be metabolites of this new organism?
Since this was mainly just like a screening project using all the funding that we had in our lab, we were just looking for something new, just any new peaks. So I'm not quite sure what could come about of this, but I'm very interested in looking
That's downstream.
at it. Yes.
So Kara, what are your next steps with these?
So my next steps with these are going to be to optimize the experiment to find a specific concentration in which we can get the most product to come out. And then from that, we will isolate it so we can run bioassays on it.
So you partially answered my question. I was going to ask you what's the quantities you expect to get from that, one, and two, how can you describe the process you will follow to scale up?
So how much we expect from these ones, LBC, we got about 11 milligrams of crude extract, so I'm expecting maybe about 1 milligram of pure extract. And then to scale up, we'll just try to optimize the experiment, and it'll just be a lot of like trial and error of finding the right concentration, because for some of them, we know it's between this, like the 10 micromolars and 100 micromolars, so we'll do that, and then we'll just probably use another piece of literature that uses fungal extracts as their basis to try to scale up our experiment.
I just want to say my father was on the line when they were making penicillin vacuums and watched in the big vats what they were doing. Very cool. And then he came back out and detailed it, which means selling it to San Francisco Hospital and bringing it out back in the 40s. California? Yeah, and so that he detailed it throughout California, and it was the only really huge pharmaceutical we had for working with patients that were obviously serious.
And that is very important, and I really appreciate you sharing that with me. But as like we all know, antibiotics might be scarce, which is why natural product research is becoming more and more important.
Very, very important.
So that is exactly what I'm trying to do here. Natural product research, trying to find new antibiotics, new antifungals, so that we can give ourselves a new chance.
Right.
Yes.
Hi, I'm Gabby Sonberger. I work with Dr. Simoshin on molecular switches. So first, a little bit of background about me. I'm from Maryland, Georgia, California, and most recently Washington State. I am a chemistry major who's currently trying to add a minor in applied mathematics, and I'll be graduating in 2023. So our research this summer was centered on the synthesis of molecular switches, which have promising applications in cancer drug delivery. So this is a model of cyclohexane in the chair conformation, and that's a particular spatial arrangement of atoms within the molecule. So on the left, the hydrogen atoms in red are positioned vertically, which is in the axial position, and those in black are equatorial. So the model on the right is the same cyclohexane molecule after a chair flip, which is a series of rotations, of bond rotations, that result in the axial and equatorial molecule switching places. So those black hydrogen atoms are now axial and the red are now equatorial. So like the cyclohexane, the molecular switches that we are synthesizing also have a chair conformation, so they can undergo that chair flip. And this change of conformation is going to impact their properties because all the groups attached to our molecule are different. So it's going to change which groups are able to interact with one another. So the chair flip in our molecular switches is driven by the addition of acid, which is going to add that red hydrogen to the nitrogen attached to the central ring. And then that red hydrogen is able to form a hydrogen bond with the oxygen next to it. And this drives the chair flip as the molecule wants to bring those two atoms close enough together to form that hydrogen bond. So Dr. Simoshin's lab has previously synthesized trans 2 amino cyclohexanol derivatives as molecular switches. And we use this information to decide which amino groups we wanted to include in our own molecular switches. So this plot shows the conformational switching using different amino groups previously tested, including this morphine group in purple and the pyrrolidine group in gray. And so the morphine and pyrrolidine derivatives are particularly promising because because of their large switching, that large difference between their starting and endpoint and that curve. And this is going to indicate that in the presence of a base, these molecules, molecular switches, are going to have the majority of their molecules with those ester tails in the equatorial position. And once an acid is added, we're going to switch to having a majority of axial tails. So because of these large promising switches, we also decided to use pyrrolidine and morphine derivatives this summer. So using these previously successful amino groups, we are altering the trans-2-aminocyclohexanol structure. So instead of attaching the ester tails at that carbonyl carbon shown at the top, we are going to be attach them at the oxygen. So the ester tails on the bottom. And we're trying to test how this difference is going to change the equilibrium in this reaction and the interactions of the cycle. This is a general outline of how we synthesized our new molecular switches this summer. And the later reactions in this scheme involve compounds that have not been published before. So we're continuing to optimize this synthesis as we work on our new switches. So for now, we've synthesized switches with benzoyl oxytails, and that is pictured on the left there. And we included both pyrrolidine and morphine derivatives with these benzoyl oxytails, as well as a selection of other aromatic and aliphatic compounds and cycles with heteroatoms to test the effects of these different amino groups. So we used the NMR, nuclear magnetic resonance spectra of these derivatives to determine their conformation. So coupling an NMR is caused by interactions between adjacent hydrogen atoms. And so when this molecule flips, it's going to change how these hydrogen atoms are able to interact. So it's going to change that NMR signal. So we looked at the coupling of hydrogen 4, which is proportional to the amount of each conformer present. So a coupling of approximately 10 Hertz, which is shown on the left, is going to indicate that there is a majority of molecules in conformation A. And you can see that it has that narrower signal that's also going to signify that. And so a coupling concept about 25 Hertz on our other side of our graph on the right is going to indicate that we have a majority of molecules in conformer B, so with those axial tails, and that's going to be a much wider signal. And so a value in signal width between these two endpoints indicates that there's a mix of axial and equatorial conformations within the sample, and we can use it to tell which is more abundant. So W observed is the coupling that is measured by the NMR, and that's what we're using to determine conformer populations for the relative amount of each conformer. So this is the coupling data that for some of the molecular switches that we synthesized this summer. And our first benzoyl derivative at the top with the morphine substituent had a coupling of 17 Hertz in methanol. And so as you can see on this graph, that's about halfway between our expected endpoints for having fully conformer A and fully conformer B. And so this means that there is a mix of axial and equatorial conformers in our sample. And so we then added an excess of trifluoroacetic acid, and that caused the coupling to change to 26.2 Hertz. So that means that we now have a large majority of conformer B. And so our next derivative that we tested is all the way on the left. That's the pyrrolidine derivative, and it originally had a coupling of 11.5 Hertz, which is very promising, as we can see that this is a large majority of conformer A. And after an excess of trifluoroacetic acid was added, we now have a coupling of 25.6 Hertz. So that means that we now have a majority of conformer B, which is very exciting, as that's exactly what we want, to go from a majority of one conformer to the majority of the other. And so this is our preliminary data on our molecular switches. So we're going to continue to work to test other switches with these same new ester tails, as well as trying to create titration curves like those you saw on the previous slides that are going to show us the exact pH required for the switch to occur. But this is very exciting because we do see at this point that switching is occurring. So one potential use for molecular switches is in targeted drug delivery. So molecular switches can be incorporated into liposomes, which are liposomes that break apart in response to an acid. So liposome, as you can see on the bottom, is made of a lipid bilayer, which is 2 layers of densely packed molecules with long tails. And a recent success of a lipid-based drug delivery system is the Pfizer and Moderna COVID-19 vaccines, which use lipid nanoparticles to encapsulate and transport the mRNA treatment. So we're working from a similar starting point, but the liposomes that we're creating would be able to be used in cancer treatment. So the Liposomes are going to be stable in neutral and basic environments because they're going to have the conformer on the left with the lipids or the molecular switches tails in the equatorial position. So they're going to be able to be incorporated into that lipid bilayer. And when the liposome encounters an acid, we're going to see that conformation change take place, and the tails will switch to an axial position and break apart that liposome. And that's going to release the drug. So that will occur at a cancerous tumor, which is if some cancerous tumors are in more acidic environments. And so that's what will cause that flip and release the drug at that particular point. And so how acidic that environment needs to be is dependent on the pH at which the molecular switch occurs. And so our future research is going to be focused on expanding the library of possible molecular switches and trying to determine these possible uses for these switches. So thank you so much to the John Stauffer Charitable Trust and donors for giving us this opportunity to do this work. Thank you to Dr. Smoshin and Oscar Mendoza.
Very nice presentation. And so anyway, questions? Yes.
Can you give us an example or two of cancers that they have this acidic environment and therefore your delivery method would be appropriate?
So I don't know the particular types of cancer, but what we're looking for is the Warburg effect, which is the result of anaerobic systems in the cancer tumors. So I'm sorry, I don't know any examples off the top of my head that have that effect, but that's what we're looking for.
How many new compounds have you made and characterized?
So we currently only have the NMR data for those two that I showed you, but we have 5, I believe, with the benzoyl oxytails right now, and we're currently creating a whole new family with benzyl tails, so non-aromatic aliphatic tails. So we're going to continue.
Okay, so I don't see any questions from audience. So okay,
Question
so
from
very
Tom.
nice and showing.
Getting a question on the Zoom. I think we were getting a question on the Zoom.
Oh, yes.
No, I think someone is trying to ask a question on
Oh,
the
okay.
Zoom.
So question, yes. Oh, yes. Okay. So Santa, could you
Not in the chat. I think they tried to unmute.
yeah, so because someone, I know someone's writing.
Yes. Yes, this is Dr. Franz.
Oh, go ahead.
Okay.
Hello?
So Gabby, you're back.
Am I good to go?
Oh,
Yes, go ahead, Dr. Franz.
A very nice presentation. I wanted to ask, in your determination of the coupling constants, were you able to do a full titration? I'm not entirely sure if that question was already asked, but do you have an idea where, at which pH approximately the flip begins to occur?
So we haven't done a titration yet. We're actually planning on doing one very soon. We only have data currently for with an excess of the acid, so the full flip.
Okay, great, because the slight acidity of cancerous tissue is critical that you hit the right pH where your fliposomes then become unstable. So, okay, great, thank you.
Yeah, so that's the reason that we're trying to synthesize a lot more or a larger variety of these different amino groups and different tails to try and hit that perfect pH.
Great. Hi, everybody. My name is Ashley Ramos, and due to having the John Stauffer Research Scholarship this summer, I was given the great opportunity to synthesize thyrodoxin mimetic peptides that underwent mass spec analysis. My faculty mentor was Dr. Ren. A little bit about myself. I have grown up here in Stockton. I swam for Tiger Aquatics here at the pool on campus. My family has taken my siblings and I, when we were little anyway, trick-or-treating here every year. I had my prom pictures here. So it was only right that I went to college here. It already feels like home, and being a member of Dr. Ren's lab has only made it feel even more like a second home for me. So I'm very happy to be here. Let's get to some science stuff. So the bigger picture objective of my research this summer was to use chemistry to understand how the active site of proteins operate, but more specifically, synthesize a series of small peptides that were to mimic and model the active site of thyrodoxin. Thyrodoxin is an enzyme that is responsible for catalyzing the breaking and forming of disulfide bonds. Associated with this process is protein folding and unfolding. Sometimes protein misfolding can occur, and the results are diseases such as Alzheimer's disease. So once we make our small peptides, they can undergo mass spectrometry analysis. Therefore, we can see different fragmentation patterns that are associated with the different sequences that we synthesize. This is going to help us understand how the location within a peptide is very important in, like, obviously its structure and then therefore its function. So the first part was to carry out solid phase peptide synthesis. There are three major steps. The first one, deprotection, then coupling, then cleavage. So we will do those steps until our peptide is completely synthesized. Which we can actually see in the top left, that is a reaction vessel where these three main steps will take place. Then our peptide is purified and put on the lyophilizer in order to prepare it for mass spec analysis, which is what we see in the remaining two photos, top right and the bottom. Okay, so once we take our sample off of the lyophilizer, it can undergo mass spec analysis. So what this is going to do is it's going to allow us to examine the different fragmentation patterns that are associated with specific B and Y ions. This tells us the location or position of cysteine residues that are within our peptides. So in the top photo, we have our instrument, and it's going to go through three quadruples. Once it is going through the three quadruples, it will go through two stages. So the first stage is called the MS stage. That's going to tell us all of the components that are in our sample. But what we really care about is the second stage, which is the MS stage. This is going to fragment our peptide, and it's going to give us an MS spectrum, which we will see an example of on the next slide. But I don't know if you can see next to Q3, there's a little gold piece there. That is called the detector, and it's going to be sent to the computer in order to give us our graph, which we will see here. Okay, so this is just an example of an MS spectrum. So the peaks here seen on, they're labeled B and Y ion. The peaks there are represent the different ions and fragments of our peptide. And here on the right, we have three peptides. So they're, the first one has the cysteine in green, it's furthest right. The middle has it cysteine in the middle, and then the last one has it furthest left. So by analyzing the MS spectrum and understanding the different fragments and the specific D and Y ion values, that we can determine that only the first peptide corresponds to this graph. Therefore, we can use mass spectrum or the mass spectrometer to determine the sequences of peptides. Okay. And also the impact that the Stauffer Research Scholarship has had on myself. I do start grad school along with Kara in the fall. So I feel like after a year and a half, not being in the lab and even just talking in front of you, this is a lot of practice. So I feel like slowly over time, I'm getting ready for grad school and taking that next step in my life. And also, I just have always had to work part-time or full-time throughout college. So this research scholarship has really done wonders for how much I can actually get done in the lab. I was able to take time off work and to spend my whole summer here, and it's been great. I also, aside from my research, have been given the opportunity to mentor other students. And that, I believe, is going to really prepare me to be a lab TA in the fall as well. And lastly, as you guys heard, I was able to use the mass spectrometer, the lyophilizer, also learn a bunch of other different techniques, learning from the other graduate students, of different ways that I wouldn't normally think how to do things, and ways that they've spent many years like trying to figure out, like learning from other people that are just super bright has been like really great for me. So I'm really hoping after being introduced to thyroidoxin and its relation in neurological diseases, hopefully do some more future research in the causation and treatment of these diseases. So lastly, thank you to the John Stauffer Charitable Trust and the donors. Thank you to Dr. Wren for spending every day of the summer with me. And thank you to the other graduate students of Dr. Wren's lab. There's Untitled, Yadender, Sheehan, and Michael. That's it. Questions? Yes, some stuff.
So from the scheme, it follows that the system fragment is the weak link in the chain. Breaks
Yeah.
always at this point. What's the reason for this? Why is it breaking at this point?
So what happens is like the misfolding, like it'll pair in the wrong place. So thyroidoxin is going to repair that. Like there are two cysteine residues that will pair with each other that normally don't. And that's what's going to cause the protein misfolding. So what thyroidoxin does is it reduces these disulfide bonds and causes to repair it.
Okay, but my question was a little different. In the mass spectrum, you have the fragments that result from breaking the chain, right, at this amino parts. And then this way, comparing two parts of the chain, you can say where is this amino acid located?
Yes.
So why this amino acid is so such a weak link in the chain? Why at this point that happens?
I'm not too sure exactly why it's at the active site is these two cysteine residues.
So I was talking about the mass spectrum. So when you're analyzing your peptides, you mentioned the sulfur could be in many different places. And you saw that the B and Y ion have different masses.
Okay.
So why does the B and Y ion break at that bond? Why is that the place that it breaks?
There are different points of fragmentation. So there are other ions besides the B and Y ions. We decided to look at the B and Y ions that fragment in these exact places. So what it is like if you're looking at B1, for example, and the point of that B1 is considering everything to the left. So the fragmentation patterns are going to be the mass to charge ratio of everything to the left, for example. So therefore the peaks are going to reflect the specific peptide.
Okay, so how about we leave the question later during the lunchtime? So basically, how the MSMS work. So I think yes. So let's continue on this. Any other questions? So now it's good. We leave question for to think.
Which is hard to do in front of the podium, so
and then the George. Yeah.
I'll throw out a question. It's maybe way far out there, but if it's important to be able to control breaking these disulfide bonds, and this relates to misformation of various proteins, could something like this yield a cure for various prion diseases?
That's what I initially had thought, but I think it's more at this point in my research to just grow an understanding of how it behaves, not necessarily like finding a cure, but hopefully someday.
One step.
Yes.
Towards that direction. Yeah.
So the question I have is like pretty much here to have a 3 amino acid peptide. Now I understand that this is the simplest version, but how does these results can be expanded to biological problems related to disulfide formation, and also how the side chain of different amino acids can affect, your outcome.
So what was important about the residues of having the cysteine where it was is that the misfolding will occur because the two cysteine residues of the active site are not, the bonds are forming where they shouldn't be forming. And therefore, if we are to synthesize like peptides that are small like this, we can better understand how the location of cysteine really matters. So using the mass spectrum is just an example to reflect how even the ionization fragmentation patterns are different. What was the second question?
It was about the side chains of the amino acids.
Answer whatever you can.
What happens if we make it anything other than sulfur containing side chain?
Oh, if I were to do something other than cysteines, well, I mean, I did do some other peptide synthesis that don't have cysteine residues in them. It was just that my research focused on cysteine primarily.
So those other residues, can they participate in disulfide linkages?
They can. So if there's a motif and it's like CXX, where XX is usually, I believe, G and C. But for right now, just focusing on the location of cysteine. But that motif usually at the active site that was shown, I believe, is a glycine and another cysteine.
All right, I think enough for today.
Hello, everybody. My name is Cecilia Tran, and I'll be presenting my research on the molecular dynamics of lithium acetate fragmentation. This was in Dr. McCullough's lab, and he advised all of this. About me, I am from Fresno, California. I'm a fourth-year chemistry student, and the reason why I chose to do Stauffer this summer is to get a head start on research before going into more in grad school. So some background information. Mass spectrometry is a widely used analytical method for determining the structure of a compound. And this has many uses, like in medical situations to identify proteins, but also in the environment for contamination analysis and carbon dating. The type of mass spectrometry associated with this research was ESMS, which stands for Electrospray mass spectrometry. And you can see how this works in the diagram here. First, the sample is charged using a high voltage and then sprayed onto the detector. As the droplets move towards the detector, they will fragment because the charge becomes too great for the droplet size. And on the detector, it kind of counts the amount of clusters present for each cluster size. And that's where a mass spectra is made. This one by Dr. Bogdanov. This is on his project on lithium acetates as well. And you can see from this curve that this is a Boltzmann distribution, which shows the general trend of how the distribution should be for an ideal gas. At n equals 12 is a really high peak, which is unusually stable, which is why we want to use this as the parent cluster for the fragmentation. And at n equals 12, it's just 12 lithium acetates with an extra lithium. And I'm going to abbreviate this as a 12 or plus. And so the motivation for my research is to use computational methods to gain more insight onto experimental distributions. And that is specifically with TDMD, which stands for temperature dissociation molecular dynamics. And when looking into molecular dynamics, the first thing to do is to make sure that the simulated data agrees with the experimental data on a physical lab. So some methods were a bunch of computer programs, including PACO, BMD, and NAMD. After determining the cluster size, which would be 13 lithiums and 12 acetates, it's put into PAC12 to pack the atoms in a defined space, giving each atom a coordinate. And then it's ready to be put into BMD to visualize the cluster and assign the bonds, and get ready for the next analysis. Here's a structure from BMD of the parent cluster, 12 or plus. And NAMD
uses classical molecular dynamics to perform simulation. And this is done in a series of stages. So first, the cluster is relaxed through minimization to make sure that nothing's overlapping or too close together. And then heating adds kinetic energy to it to get the fragmentation going. And then equilibration overall to make sure the temperature and gas control. Following NAND, the data that we get is the cluster size and its abundance. And from this, I made some mass spectra. This one's taken at 1100 Kelvin and 10 nanoseconds. And you can see that this is still a really high peak at 12, showing that the parent didn't fragment as much. But you can see going down from the highest peaks, there seems to be a pathway from 12 to 10 to 8. And I took more data at 1200 Kelvin, and you can see that it's further along, but the parent like with a lower abundance now. And the most important thing to note here, this is just a comparison between the two spectra. That there is a lot of skipping going on, where it goes from high to low to high on both sides. And this is important to note in the findings, since the skipping shows the neutral dimer as the major product. It also correlates with experimental data, with my data at the simulated point on the left here, and the experimental on the right, and both have a really high abundance of the Tetomerase plus. So it's like leading to the same outcome. And this whole thing shows that TDMG can be a new method for counting neutral molecules, since mass spectra only shows the charged species. So from here, I just want to generate more data over more temperatures, and then perform kinetic studies to see why a certain pathway is seen. I already got started on this, that's something else. And maybe look into higher charge clusters to see how they undergo fission, since it's more complicated with higher charges. So that concludes our presentation. I'd like to thank the Charitable Trust and donors, Dr. McCullough, and the European Chemistry Department.
All right, very nice presentation. So now questions. All right, Andrew.
I have like 2 questions.
All right, you can.
My first question is, what was like your sample size on this? Was this like the triplicate, or like how many simulations did you run?
So I ran 100 simulations because it takes like a few days for each one. I don't want to do too much.
How long were they?
They were 100. Oh, like how long did it take?
Yeah, no, like how long was each like simulation?
I'm not sure how long. It takes like an hour for one. So if I did 100, it'll take like 2.5 days.
Okay, like in like nanosecond, what's this game? Is this in vacuum? Is this like in a vacuum, or is it like solving?
Oh, it's supposed to be in solvent in a aqueous solution, but I did mine at a certain like cube. I forgot what it's called, but as the molecule moves in the cube, it kind of just keeps it in that space. So it just moves right back on the other side.
Other questions?
Question from Zoom, please.
Yes, we can hear you. Dr. Franz.
All right. Yeah, thank you. Cecilia, do you remember or do you know what method you used to carry out the molecular dynamic simulation? Was this a plain force field, or did it involve quantum mechanical, like orbital overlap components? What did you use?
I just used a bunch, or I used a file that was given to me, but it just uses classical dynamics, so just applying kinetic energy, and it follows Newton's second law of motion.
Thank you. Maybe Dr. McCullough can clarify this. She is just beginning to learn, so the question to think about the field.
Did Andy ask about the force field?
What program?
It's AMD uses Charm.
Sure.
Thank you.
Okay, so any questions? Alrighty, I think we can move on. Alrighty, Cecilia.
Hello, everybody. My name is Guy Lawson.
My name is Kim Harvey.
And together we're presenting the testing linked inhibitors of the ubiquitin cascade. But before we get into that, a little bit about each of us.
So my name is Kim Harvey. My hometown is French Camp, which is a small town really close to Stockton. I'm a biochemistry major, and I'm a senior, and I'll be graduating this fall. I chose UOB because it's a really great university that's only 15 minutes away from my home. I chose to do the Stauffer Summer Research because it would give me an opportunity working in a real lab doing real science and would give me some practice to later move on to graduate school.
And hello again, my name is Guy Lawson again. So my hometown Stockton, my major is biochemistry and I'm a fourth year at UOB. And the main reason why I even came to UOB was because, first of all, it's pretty familiar, so that's nice. And not only that, it's a great place to study medicine. So on top of that, the reason why I wanted to do the Stauffer was because it kind of gives you an insight as to how like a lab is actually conducted rather than just far away experience from a book, right? So specifically in this lab that we did, we're studying this protein called ubiquitin. So what is ubiquitin? Ubiquitin is obviously protein, and it's involved in this thing called PTM, Post-Translational Modification. So what that means essentially is that the ubiquitin in the body can essentially flag proteins for degradation to the proteasome. So that essentially just means that it can just flag a protein so that it can be marked to be broken down into its constituent parts. And this is very this is a very important function, and it's implicated in numerous different diseases as well as therapies for.
So in our lab, we're interested in stopping proteins from being tagged with this ubiquitin. And in order to do so, we need to target the mechanism by which the protein gets flagged with ubiquitin. This mechanism is called the ubiquitin cascade, seen here. And it consists of 3 proteins, E1, E2, and E3, that each pass the ubiquitin essentially off to the other until it is finally chemically bonded to its final protein. In our lab, we're mainly interested in targeting the E2 protein, which is right in the middle of the ubiquitin cascade. And we're interested in doing this by designing small proteins, which are called inhibitors, that will bind to the E2 and thus effectively keep it from being able to interact with other members of the cascade and thus shutting this whole thing down. There are actually 30 different types of E2s that have been identified in the human body, and not much is known about these, but we're interested in designing inhibitors for essentially each one of these.
Speaking of designer proteins, specifically what we did in this lab is you can see these 2 things here, the UBL and the ring protein. So these are normally found in the human body by themselves. But what we did in this lab is we essentially attached them together. And what this essentially means is that it can bind like a Velcro glove, like over to the sides, not specifically on the E2. It will bind and it will block the receptor. So this has very many useful implications and can essentially stop the cascade pictured on the slide previous. And not only can it stop the cascade on slide previous, it can also prevent the cascade further down because of the way that the protein is bound. It is not bound directly on there, and it kind of blocks other biochemical mechanisms that could potentially finish the rest of the cycle. And this specifically is very, very cool in my opinion, because the ring and the UBL protein are based on different E1s that actually bind to E2 already. So this is really like an example of biological engineering.
So a lot of the work that we just described has been done previously, but the goal over this summer was to determine the specificity of the 2 E2 inhibitors that we designed, the RU and the UU inhibitors. Specificity means that they interact well with some of the E2s, but not with all 30 of them. And we did experimentally determine that they do work well with some of these and not with the others, but we needed to do another test that just focuses on the protein-protein interactions to kind of double check these results. And this biochemical test is called the Yeast 2 hybrid screening. So essentially, we put our two proteins we're studying, the E2 and the inhibitor, inside of yeast cells. And these yeast cells are genetically modified to where they are unable to produce their own histidine. And histidine is one of the an amino acid. And of course, because it's one of the amino acids, which are the building blocks for proteins, histidine is a vital nutrient for the yeast cells. So if the two proteins inside the yeast interact with each other, the yeast will be empowered to actually make histidine and therefore would be able to thrive in an environment that did not have this amino acid. However, if the two proteins inside the yeast did not interact with each other, the yeast would not be able to make histidine and would die in an environment lacking histidine. Therefore, we use the amount of yeast growth in a media that lacks this one nutrient to assess how well our two proteins interact with each other. So essentially, if we see yeast growth in this histidine lacking media, this is a positive result, and it means that our two proteins successfully interact with each other. If we see no yeast growth in the histidine deficient media, this is a negative result, and it means that our two proteins do not interact with each other.
Okay, what you're seeing here is the graph of the results. So specifically, my project was UU and Kim's was RU, right? So what you're seeing here in these three columns is the negative control, the positive control, and the actual experimental trial. So the SD alone was negative control because, if you don't give the H or the A, even if the two proteins conjugate, it'll create the H, but there's still not going to be A, so the yeast won't grow ever. And for the second, for the SD plus HA, if you just give everything the yeast needs to grow before, you even run whether or not it's going to be, the proteins are going to be conjugated, the yeast are going to grow regardless of whether or not the proteins conjugate. And for the actual interesting experimental trial, it is SD plus A. So this is very useful because if the two proteins conjugate, then we know that there's some interaction with the inhibitor, between the inhibitor and the E2s. So as you can see here, the experimental columns for the UB2E1, UB2E2, UB2E3, they all interact, and that shows us that there's there's interaction between the two proteins, so there's likely something there. And for the UB2G1 and the UB2H, they do not. So that shows us that there likely isn't interaction there.
So for the RU results, we also see some interactions with the UBE2 E's and very little to no interaction with the UBE2G1 and the UBE2H. However, the RU does show reduced activity with the UBE2E's compared to the UU, which means the UU has the greatest specificity for the UBE2E's.
So what are the practical implications of all of this research, right? So let's get back to the beginning. What is ubiquitin? Ubiquitin is essentially a protein that flags other proteins for degradation. So this has numerous great implications for potential therapies involving many different diseases, such as cancer, Alzheimer's, and pretty much everything listed up here. But specifically in cancer, you have the rapid and unregulated proliferation of cells, which, if you have dysregulated cellular mechanisms, it might be useful to flag things for proteins for degradation. And in Alzheimer's specifically, you have amyloid plaques in the brain, and those can't be degraded. So ubiquitin could potentially be involved in useful therapies for treating some of these diseases, and potentially understanding in this lab aims to understand ubiquitin better by discovering the specific chemical what mechanisms chemically make ubiquitin so make it inhibit. And on this slide, you see kind of what we did in lab. On here, you see the different yeast plates, which we have to pour. And then here you see the stalks of the colonies that were picked of the viable. And you see the reagents.
And we also see that we had to also in this lab purify proteins and then look at the results on these gels and centrifuge cells, do all sorts of other things related to the research.
And overall, what I learned most about this lab, especially from the techniques used, were first, with the logistics of actually running a lab, right? There's like a million things that you don't think of. You know, you need to order things, you need to store things. And these are not things that you really think of usually when you think of lab work. And not only that, I learned about yeast 2 hybrids as well as different biological methods as for actually conducting research.
And I learned essentially how to do good quality science work, including the controls, having a good, clean, sterile work environment, and keeping track of the multiple projects you're running at the same time. So in the future, in this lab, we're going to just continue doing more work and more growth assays with the rest of the E2s that need to be studied. And in the future, I will be moving on to graduate school at UOP. I'm probably going to do a PhD in biochemistry. And then I'm interested in later beginning my career as a biochemical researcher.
For me, I'm looking to go into medicine, specifically PA, but I wouldn't be opposed to being a researcher either. It's very, very interesting. So overall, thank you so much. Thank you, John Stauffer Charitable Trust and donors. Thank you so much to Dr. Harrison, as well as thank you so much to the graduate students that helped us so much, Lee and Rose. Thank you.
Such nice dual presentation. So let's do a short question. Bob.
I'm curious, I couldn't see all of the applications. I can't wear a mask and glasses at the same time. But would some of this work ultimately be applied in treating autoimmune diseases like rheumatoid arthritis or something of that nature?
I could potentially see that, yeah, because so proteins are involved, especially like so rheumatoid arthritis, if I'm not mistaken, it's essentially inflammation of the joints, right? So that usually is involved. It's usually something to do with inflammatory cytokines. So cytokines are just another protein. So that ubiquitin research could potentially help and aid in develop certain research or certain therapies in order to treat rheumatoid arthritis. Yes.
Or lupus erythematosus and several others too.
If it's involved in protein degradation, then there's a high chance. Like ubiquitin is found in quite a few areas of the body, so potentially.
No questions? Alrighty, so I think everyone's hungry. Okay, let's thank so I think we're going to close this symposium. You see that, right? How great our researchers, they are staying here just for the summer of the in-person. Now, how much you have learned. I'm so impressed myself. So I think I also thank the faculty mentors dedicated to mentor them. And each of them, you see their presentation also mentioned graduate students. I thank the graduate students in all the faculty research labs program. And also thank all the faculty or students and also universities supporting. That's incredible. And last but not least, the donors. Thank you so much for making this possible. And so last one, I'm going to, yes, you see this short list that donors, they have direct connection with Pacific. So yes, you can reference yourself. And so but I think today we'll go for a lunch, we talk, and then you have any questions to ask, any suggestions, you let us know, and we'll make this even better. So thanks for the fundraising team and amazing work. So, okay, now
You've joined on Zoom. We appreciate your participation. It's recorded, so we will go ahead and make sure that we get this presentation out to everybody who is on the Zoom. and anybody else in the audience who would like it. So if you have family members that couldn't attend, just let me know and I'll make sure that we get the recording out. So we appreciate everybody joining us today, and we look forward to seeing you at the 4th annual Stauffer Symposium next year, which hopefully will be 100% in person and live. Yes, thank you everybody on Zoom. Thank you.